Hello everyone, my name is Bob Harveson and I'm an extension plant pathologist at Scottsbluff, and I'm here today to talk about specialty crop diseases and some of the projects that we've done this season on trying to find ways of reducing disease problems. The specialty crops that we're going to be talking about today are the sugar beets, sunflowers, dry beans, chickpeas, and lentils. I'm going to start off here with the sugar beets. Well, first, before that, I want to talk about the hailstorm. That was the primarily problem for everything this year. A huge storm came through on June 29th, and it affected every single field study that we had. Some of these we replanted again, others we destroyed, and there was even a few that we just let go and just see what happens. In terms of sugar beet diseases, there's two major ones that we most commonly find each year. They also are considered to be the most problematic. Therefore, we do often have research requests to do studies that involve those two pathogens. Begin with Rhizoctonia root rot. This, and you'll see up here in this corner, this is the symptoms. The plants wilt and then just fall apart. They're right beside it, just hit the ground. This is what the roots look like when they've been highly diseased with the lesions. They're the dark structures. Cercospora leaf spot is obviously a leaf disease, foliar disease. And the little circles that you see here are lesions where the pathogen is located, and spores are produced and they're moved around through water or wind, and then if they hit a spot on the leaves that are wet, well then they germinate and cause more problems, and this can go on and on and on till it looks like this, where you've got a whole bunch of them that have taken the life out of it. This slide is one that's depicting one of the sugar beet trials, and this one up here in the top was showing you how well the crop looked like in the 25th of June. The thing was planted in May, but then it came out in late May, and then a month later, this is what it looked like. So this was really useful, but unfortunately, on the 29th is when that storm came through, and then this is what the field looked like after the storm had gone. But we decided not to replant or anything like that, or start over again. We just let it go. And so this is what it started coming back. It wasn't killed, but it just took away all the leaves. And then this is what it looked like in September, right before harvest. So it was enough to at least get a harvest value, which I think was around 18 tons per acre. So that wasn't real good, but it was better than digging it out. And the next one is the sunflower disease issues. And over the last few years, we've been there's two people from North Dakota State that I have got together with, and so we all do the same projects in the different locations where we are. And Fabina has taken care of the Phomopsis in terms of leading that. Sam has taken the lead on the rust, and then I've had the Rhizopus head rots, which you have finished and cleaned up. Well, this is a picture of the disease symptoms. Phomopsis is a fungal disease that originally starts as as a leaf infection, and then it goes and attacks down the petiole into the stem, where it causes these lesions like this that eat away the contents inside the stem, which causes them to become they can't hold up the weight of the heads, so they fall over like you see here. And that's where the loss comes from, is that those are not picked up by the harvester even when they fall over like that. So that's the symptoms of this, and it's, like I've said, it was really considered to be the worst problem for sunflowers currently in U.S. and Canada. This was, we had several different projects, but this is the one that we had where we were looking at different chemicals and looking at different varieties of a susceptible and a resistant one. Again, this field just looks, the couple days after the storm, that's what it looked like. But in this case, we decided to replant, and so it was a huge study, and we had to replant it again on the 2nd of July, and just took it to form. But we did not get any yield data on this. It was just planted too early, planted too late, and but we did get disease values from that, so that we got a little bit of data, but not very much. Sunflower rust. This is a disease that we 15 years ago was a severe problem, and it kind of went away, but for some reason it has been emerging again in this area. And so this is a study. Here's more symptoms of the spores are on the back of that leaf. These are just 2 leaves that are exhibiting the symptoms and of the pathogen. The spores are locked up in that, and this is how the pathogen will overwinter. Okay, here's the study we were doing, and this is where they wanted to find out from the USDA whether they had any new breeding lines that had any rust resistance. So that's what this test was, and it included almost 300 breeding lines that with 2 reps per treatment, we were able to, it was really difficult as well. It's 588 total plots. These were one row plots, and we planted them by hand, which really made it difficult to do this, not having the machinery. But anyway, this is the next study that we did. Here's after a couple of weeks. If you look down in here, it is there's some of these that emerged really well, and others that did not. But this was several weeks after we planted. This was something that when we got to the end of the season, this just showed what the breeder was trying to do to find out if there's any of his lines that had any kind of resistance. But this particular one shows how this poor thing did not have any resistance to the disease, so it really was a severe problem that we decided not to replant this one because we didn't have the seeds. Now, moving over to dry bean diseases, these are the 4 common diseases that you will see here in Nebraska. It's brown spot here, wilt, common blight, and halo blight. Well, just the slide previously showing the different pathogens of beans doesn't always look like that. What they also, more often than not, will look like will be multiple beans in the same infection. So this is This caused us to think about this as just one problem. We didn't worry about which disease it was, because they all have about the same thing required for managing. So that we took it that way. And based upon that information, since 2010, we've been conducting studies that focus on bacterial diseases, and we don't care about which one it is, with various products. We did begin this thing with copper, focused on bacterial wilt, but it was not very successful. We do know that copper products work sporadically. Some of them are more sensitive than others, and so that's why we tried to start looking at other products. Based on the information that we acquired, we used a lot of different products to test, and this is just a list of those particular ones, ranging from copper to fatty acids, hydrogen peroxide, Bacillus species, which is another bacterium, and this is just a list of the ones that we did. Okay, we found that by 2017 we felt like we had done enough work to find some products where we saw some positive effects to manage the results on dry beans. In fact, it was good enough that we were able to publish the article, Improving Yields and Managing Dry Bean Bacterial Diseases in Nebraska with New Copper Alternative Chemicals. And the ones that ended up being the best, that always led to the top, was this EcroAgra A300 and then Sanidate. So both of these products were the ones that worked the best, and now I know that a lot of people are moving to those hydrogen peroxide ones, the Sanidate and Oxidate. Based on the success of these other products, we decided to see if these same products would have any effect on fungal diseases such as white mold or rust on dry beans. And then we also are still doing this, but we were trying to determine whether these copper alternative products could work on other pulses such as chickpeas and lentils. Next up is the dry bean root rot diseases that we may see. There's quite a few of them here. There's up to 5 that I've seen before, but the 2 that we're just really concerned about is Fusarium root rot and Rhizoctonia root rot. This is what the Fusarium looks like. It's just these lesions on the bottom here just begin to get worse, and this is one that this went completely to harvest, but notice how the root was killed exactly. But the plant was able to stay alive by producing all these other little roots coming out, but it still was severely infected. Rhizoctonia looks different a little bit than Fusarium by producing these lesions that are sunk down a little bit. But if you open the stems, they have this brick red material stain in the inside. So that's something that's diagnostic for Rhizoctonia in dry beans. One of the problems with these root rot diseases is they're really difficult to identify. They wilt like this, but a lot of other problems could be wilt like this. As a matter of fact, this particular slide was bacterial wilt, but this is what it gives you an idea of the difficult on getting the right pathogen is causing the problems. Other issues with working with root rots is that they require different fungicides. The ones on Pythium are not going to work for the ones on Rhizoctonia, so you need something different fungicides for different problems. That's again why you need to correctly identify what the problem is. They also were not a problem every year. Even though they are right there in the soil, they might not use, they can live for years, but they might not be a problem every year. And then once you do notice that it is, it's often too late to do anything that year. So that's another problem. It's difficult to reach in the soils like that. And so basically the most cost-effective measures that have been found is genetics, breeding, and seed treatments. And since Carlo, our bean breeder, is taking care of that, the seed treatments we attempted to try by, we were asked to do this by the Dry Bean Commission in 2023. And as that time, we've been conducting using various fungicides as seed treatments. And unfortunately, we haven't learned a whole lot. There's not many differences among the treatments for 2 years, and then probably the reason why is there just wasn't any low levels of disease, so we didn't see any differences in the treatments. But the thing the problem in 25 again was the hailstorm, and that again was replanted on July 2nd, and this is one that we did try to take to harvest. And as you notice here, it's very late, and because of it was so late in planting, the data was poor, but just for good faith, we still tried and took it to heart. Okay, now to round this out, the next group of crops that we're going to be looking at is chickpeas and lentils. I have had studies, we've done studies on cowpeas and dry yellow peas and mung beans, so on. But right now, for today, I'm just going to be talking about the chickpeas and the lentils. Chickpeas are also called garbanzo beans. Now, I've been working on this with Carlos, the Uria, our bean breeder, quite a lot. We've been working on this for a long time, and just found that that this is the major disease problem that chickpea has. It is caused by a fungus, Ascochyta rabii. There's also root diseases sometimes, but not, and it's by a whole complex of different ones. So we really don't know exactly on that. But so we're just really spending more time looking at the Ascochyta blight than with the root diseases for chickpeas. But right now, this is a major problem. If you look at this, this is at harvest or at the end of the season, and all those circles and so forth on the pods are just that's Ascochyta. So it's really done serious damage. It's this thing can occur on the pods, like you see here, or the leaves or the stem. So it can hit any above ground tissue. This is the breeding plots with Carlos, and just different ones that he's making selections and trying to create new cultivars. And he has released several that do have good resistance, but there still is a need for fungicides in addition to breeding plots. But what it does is it removes the number of applications would be needed for this, along with the genetics. Now, as I've said, we've been working on this for a long time, and there's a number of things that we have for options and what we are using and testing. But breeding is probably the best that we're going to do, but it does take time. So in that time period, while we're waiting for these things to come out, there's other options we have. There's chemicals. There are some fungicides, but the problem is that some of those isolates that we have in this area have developed resistance to some of the sterile urine fungicides, like Headline or Quadris. So that's something else that kind of makes it difficult. So what we're going to do, or what we are planning on doing, is to test those alternative chemicals that we did with dry beans, and test them to see if they have any way to anything to help with the problems like they have with the coppers and bacterial diseases. And we did have a project in 2025 from the Pea and Lentil Crop Commission, but just like every other study, we had a train wreck. This one was actually planted 3 times, and we still couldn't get a good and did not go to harvest. So we tried and tried, but this one was a mess, and just plowed it under, and not going to worry about it till next year. Lentils. This is a new thing, and this is what we have in a new USDA proposed or study. And what we started doing in 2025 is going ahead and planting, and we had a really good stand coming up. It was looking really good, but then after 2 days after this picture that we see up in the top corner have been, it's something got it with herbicide drift from somebody in the neighborhood. So that was a problem, and then here up in the top right, that it also was completely hammered by hail. So we did replant in another area due to that. We planted another area so we could get away from where we had the issue with the herbicides. But that one was unsuccessful just because of in that point it was planted so late that there was too much heat. It couldn't tolerate that, and we had problems with the well for the irrigation. So with those 2 problems, again, this was just we had to just throw it away and just it's another wash. But I'm going to follow up here with this is something that's kind of been working. It's sort of a side project. It's fun, and it's not going. We're not going to have a problem with it being a economic issue. But this is a little study that we were doing. If you look at the 2 pots on the outside, and there is some of those leaves are showing symptoms of bacterial wilt, and then the one in the middle, those plants are just absolutely sloshed. The one there was the control. It's still there, but what the deal is that these particular isolates, although they were pathogenic, were not nearly as virulent as the one in the middle. That was one of our standards here, and those actually were 2 that were recovered from the stratosphere. And so that sort of thing just kind of opened the whole door up for us. In 2023, I was asked to join this group of microbiologists on characterizing wilts, or what they thought were wilt isolates, but they had gotten these from the stratosphere. And so I don't know how they got down. They just fell down in certain ways. But the thing people were worried about is that, oh man, a new one. How are we going to handle this? And the tests that they were running, there was a number of those. And then I sent 15 or so of our isolates that we had collected over the last 2 decades. But the point is that we had some species, we had some isolates that were identical to several of the ones from the stratosphere. So what that is it was not nearly as virulent as the ones that we have already. But what that, it's probably a so what type of thing, but it's still what it says is that those isolates from the stratosphere were different. They were different enough from Bacterium flocculum faciens to be a different new species. And so as being a part of that group, it's been kind of fun and so forth to be a part of that where a new species was made. And it also, to me, suggests this is the interesting thing to me. It suggests that this pathogen's been around for a while because it was initially discovered and collected from a millet field in 2007, and it was just in our freezer. And now that particular isolate was the exact same as one that came out of the stratosphere. So anyway, that's just a little by the way thing. But anyway, any questions? I'm happy to answer.